All right, so how does your DNA actually tell your body what proteins to make? How does protein synthesis really work? So remember how your DNA was like a series of letters, could be A, T, C, G, 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 C, C, T, A, G, C, C, T, A, A. Okay. And it goes on like that for 3 billion letters. Okay. And it might be that one section of this, maybe from like here to here, codes for your eye color. And then maybe another section codes for your hair color, and another section codes for your intelligence. What happens is this is the code for what, let's say this is eye color. This is the code for what your eyes should look like. Okay, so how does that code really tell your body what to do? Well, first of all, if you remember RNA, instead of T, wherever you see a T, you change it to a U. So first of all, we have to address that, is that your DNA is so precious inside the nucleus of your cell, and you don't want to damage it because that's how cancer starts. So you never want to damage your DNA. So what we do is we leave the DNA right in the nucleus. We don't take it out. It would be hard to get out anyways because it's huge. And what we do is we make a little copy of it, and that's called messenger RNA. It's like it goes, makes a copy, and then it's like acts like the little messenger. It runs and it brings that code. All right, so let's say your messenger RNA went into the nucleus, said, you stay here, DNA, you're too important. I will take this message to the ribosome to make protein. And it takes the code. So let's say that your code was UAU, UGG, CAA. All right, so that's your code for your protein. It would be longer. So then it goes and it looks it up. It goes, okay, UAU. So it looks here and it says, okay, oh, UAU, that means the first amino acid in this protein should be tyrosine. And then UGG, says, oh, tryptophan is next. And CAA codes for, where is it? Glutamine. So this code in your DNA is literally telling your body which amino acids to put together to make that blue-eyed protein or whatever it is it's making. We call these little codes codons, and as you can see, they're always like sets of 3. This chart is a little bit easier to read the codons. So instead of just like randomly looking for, okay, where is AGU? Like I'm just looking all over randomly, where's AGU? I can say, okay, the first letter is A. The second letter is G. So it's got to be here. And then actually the last letter would be here. But by the time I get to the right square, then I'm just like, oh, there's AGU. So that helps me to go, oh, AGU is SER, serine. So if we were in class together, you would get good at reading these codons. We would play codon bingo, where you have to like translate the codons to the right amino acids, and it would be fun. But we're not in class. All right, so let's go over the steps of how this process is actually done. It's got kind of 2 major steps that we call transcription and then translation. So if you're transcribing something, like a person that's transcribing, like if someone's writing your notes for you, they're transcribing, or court stenographer like transcribes, writes everything down that happens in court. But anyways, transcribing is like the part when the little messenger RNA goes into the DNA and it's like, excuse me, DNA, I'm just going to make a copy because you're too important. You need to stay in the nucleus. I'm going to make a quick copy and bring it out to the ribosome so that we can take your directions and go make the protein that we need. So it's kind of like if you had this big recipe book, the DNA is the recipe book, but you only wanted the recipe for blueberry bread, blue eyes, you would go in and like just write down that recipe and then take that piece of paper with you. Like you couldn't take it out of the library. You can't take the DNA out of the library. So you just transcribe that one recipe for that one protein you want to make, and you go and take it to like your kitchen where you would make the protein. In this case, the blueberry bread. In this case, it would be the ribosome is where proteins are made. So the steps for this are step 1, unzip the DNA. So if you remember, DNA is like shaped like this, and those A's, T's, G's, C, there's more, but they're like written on the inside. So first step is you just need to like unwind it and unzip it. Okay, let me take those 2 strips and just like untwist them. And then I really want to kind of open up those 2 strips so I can see, because there's like maybe here there's an A bonded to a T, and here's a G stuck to a C. So I just want to like expose those. So I'm going to like unwind it, and then unzip it, kind of open it up so I can see those A's, T's, G's, and C's. Okay, so let's say I've opened up my DNA. It's like kind of open, and I'm just kind of looking at one side of it, and that one happens to go A, G, C, T. And I'm like, okay, I'm the messenger RNA. I need to make a copy and run and bring it to the ribosome. So that's going to be me over here. I'm going to be the messenger RNA. And the way I'm going to make a copy is by like doing the complement to that. I'd say, okay, complement to A is T, except I'm RNA and RNA can't use T. So I'm going to do a U instead. And then another U to go with this A. Go with C's, C goes with G, T goes with A. All right, so now I've kind of made a copy, but it's like a complementary strand. And I use the original to kind of tell me what to make. Okay, so now that I've made this copy, I'm ready to go make my protein. I've got the recipe. I need to bring it to the kitchen. The kitchen is a ribosome. That's where proteins are made. So if you remember like the endoplasmic reticulum, remember how it was called like the rough ER because it had those dots on it? Those little dots, those are ribosomes, and that's where proteins are made. And the way I always remembered that is because like the littlest little tiny dots make the strongest, most important thing, protein. And the second major part of this process, so we did the transcribing, we wrote down the recipe, the A, T, G, U, whatever, the whole recipe. So here is the recipe that we wrote down. Here's that messenger RNA that we went and transcribed. So we wrote down the recipe, and now we've got to put together the right things to make the right protein. Now we have to like translate what this all means. We have to say, okay, our first little section, our first little codon, which you'll notice is 3. Our first little codon is UAC. So I have to translate that and go, okay, what does UAC mean? Let me go back to that little chart and look up what it means. UAC, I got to translate it into like what amino acid that actually translates to. So because I'm literally translating this code, I'm going to call this step translation, turning the code into making something. So that code's going to say, okay, that is serine. So we need to go fetch the serine. Somebody bring a serine. And that serine is going to have the matching code. So it's like, okay, messenger RNA, if your code is UAC, then the complement to that code is UAG. Remember how that's always like U goes with A goes with U, G goes with C. So if you didn't follow all that, that's okay. Basically, you have to know is that the messenger RNA has got a code, and it's going to call for certain amino acids to be delivered. Like, okay, the first one is serine. Okay, we need some serine. So the last thing we have is these little transport RNAs. So what do? They transport things. This messenger RNA goes, okay, I got a UAG. I need you to get a serine. Bring over a serine. So it goes, oh, okay, I can do that. And it brings over the right amino acid. And then it says, okay, next one I have a CCC. And that codes for tryptophan. So this thing goes and transport RNA goes, okay, I'll transport it. And it brings in the tryptophan. And then the next one says, okay, I have an ACU. I need some glycine. So then the little transport or RNA goes and gets that and goes, okay, we need a glycine. And what happens is you just keep the little mechanism in the ribosome keeps reading the code, translating it to get the right amino acids, and it's putting them together to make the right protein. Okay, so if you didn't catch every little detail of that, that's fine. We would have spent like a week on this in class, and I'm just giving you the broad picture. So let's say that just to conclude, what you really need to know is that from the first video, genes direct protein synthesis. Like you knew your genes made you have blue eyes, but you didn't know how. Now you know it's that the genes told your body how to make the blue protein that goes in your eye. Okay. And there are two steps to that. One is with the transcription, where the messenger RNA transcribes the code from the DNA. It says, okay, let me write this down. I need an AUGCUUCA. Okay, that's where the little messenger RNA goes in and says, okay, I'm not important enough, but DNA, you're super important. You're super big. You're the king. You need to stay in the nucleus and stay safe. I will transcribe, I will write down your code for this one thing, this blue-eyed gene, and I will go and bring it to the ribosome to make that protein. Okay, so two steps to this. Transcription is where messenger RNA writes down that code, and then translation is where it gets translated into actually making a protein. It's where you go, okay, the first part is AUG, okay, that's serine, and then, okay, the next part, and I'm making that up, I don't know if it's serine, the next part is CU, okay, that's glycine, and it starts translating to put together the right amino acids to make the right protein. So one more time, genes tell your bodies which proteins to make. How do they do it? They do it by transcribing the code and then translating the code in your DNA.